Next, very important kind of vectors are bacteriophage vectors. Now, what are these bacteriophage vectors? What is bacteriophage? Bacteriophage is a virus which infects bacterial cells. This has a property, this bacteriophage virus has a property to infect bacterial cells. So, bacteriophages are the virus that infect bacterial cells after uh, injecting their genetic material and kill them. Means, for example, if this is a bacterial cell, and there is a bacteriophage virus. That bacteriophage virus, it inject its bacteria in a bacterial cell, and after that which injects its it injects its DNA in the bacterial cell, and in the bacteria we'll get many copies of this bacteriophage virus. And after that, when this cell gets infected, lysis of the cell happens and these viruses come out of the cell. So these are bacteriophages which kill the bacterial cell. The viral DNA replicates and expresses in the bacterial cell, and as I told you, a number of phage particles are produced in the bacterial cell, and they released after bursting the bacterial cell and kill the bacterial cell. This is known as lytic cycle of bacteriophage. Means this is the process by which this bacteriophage virus works. Now the released phages reinfect the live cells. Means if there are other cells near these bacteriophage viruses, they infect these other cells also. Now, this property of transferring the viral DNA from phage capsid specific bacterial cells give insight to the scientists to explore bacteriophages and design them as a cloning vectors. When scientists saw that these bacteriophage vectors have a property to insert their DNA into a bacterial cell, and after that, through this DNA only, these viruses can infect other cells also. So they try to use this particular DNA of bacteriophage virus as a vector. So they tried and then exploit bacteriophages and designed them as a cloning vector. The two bacteriophage viruses which we have to study in class 12th biotechnology, these are phage lambda and M13. These are two bacteriophage viruses which were then modified and used as a cloning vector. So one by one we'll study about these two bacteriophage virus. One is phage lambda, another one is M13. We'll study these two viruses in our next session because I think that now, first of all, you have to learn about bacterial plasmids, PBR 322, PUC 19, TI plasmid, shuttle vectors, and then expression vectors. After that, in our next session, we'll discuss about phage lambda and M13 vector. Thank you.